Hello, and welcome to the CLSI Future Focus Forum. It is my honor and pleasure to introduce our moderator for today's program, Dr. James H. Nichols. Dr. Nichols is CLSI's president-elect. He is also professor of pathology, microbiology, and immunology, and medical director of clinical chemistry and point-of-care testing at the Vanderbilt University School of Medicine in Nashville, Tennessee. He is board certified in both clinical chemistry and toxicological chemistry by the American Board of Clinical Chemistry. Dr. Nichols spent several years as Associate Director of Clinical Chemistry, Director of Point of Care Testing, and Associate Professor of Pathology at Johns Hopkins Medical Institutions. He later served as Medical Director of Clinical Chemistry for Bay State Health in Springfield, Massachusetts, and was a professor of pathology at Tufts University School of Medicine. Dr. Nichols' research interests span evidence-based medicine, information management, laboratory automation, point-of-care testing, and toxicology. Dr. Nichols, please go ahead.
You're on mute, Jim.
Thank you for that introduction. And it's my distinct honor to be your moderator today and part of this Future Focus Forum. I want to provide you first with a welcome and overview of CLSI. Next, we'll get into the panel discussion, where we'll hear from Dr. Ren Salerno on the government perspective. Next, with Gary Prokop on the healthcare perspective. And we'll end with Dr. Darlene Solomon on the industry perspective. We'll have time for questions and answers, and I'll be soliciting those throughout the program from our attendees. Next slide. After this session, you will understand CLSI, the Clinical Laboratory and Standards Institute, who we are and what we do. You'll learn about future directions for laboratory medicine and discuss how CLSI fits into that future state. Next slide. If you have a question, be sure to enter it into the Q&A box at the bottom of the Zoom webinar. We'll be asking for some feedback as we go along, so please enter your feedback into the chat box. Next slide. Who are we? CLSI is a standards development organization that creates global best practices for medical laboratories. We facilitate the development of standards using an open, transparent, consensus-based process. We aim to help medical laboratories meet accreditation, regulatory, and public health requirements. Most importantly, we strive to improve patient care by facilitating the development of standards and products that will enable laboratories to provide the highest quality of medical laboratory testing. Our membership is both part of the professions, the government, as well as the industry. Next slide. Part of our future CLSI strategic goals is to have a vision of setting the standard in laboratory medicine for a healthier world. Our mission is to develop clinical and laboratory practices and to promote their use worldwide. We define and understand our member and customer needs. We want to create products and services with high market relevance and impact. To strengthen CLSI's global positioning and leadership, and most importantly, to be a thriving organization where our volunteers find participation relevant, rewarding, and valuable. Next slide. Here's a profile of our membership. We have more than 24,000 individuals with membership from 75 different countries. Those are divided into hospitals and independent laboratory organization, health systems, government organizations, and industry. We have more than 1,200 active volunteers who are developing our guidelines and standards, and more than 400 individual members that are participating in CLSI. Next slide. We have a global partnership of standards in action to provide targeted technical assistance in 22 countries through multiple languages and translation of our standards and guidelines. We support more than 61 laboratories and have helped them to achieve and maintain accreditation aligned with international standards through the technical skills promotion of quality management systems, quality control, method evaluation, molecular, point of care and rapid testing, as well as microbiology. And we've assisted with ISO standards implementation in these facilities. It's truly integration at in-service and pre-service levels. Next slide. Some of our CLSI products. Next slide. We have a variety of specialty areas in automation and informatics, clinical chemistry and toxicology, hematology, immunology and ligand assay, method evaluation, microbiology, molecular diagnostics, newborn screening, point of care testing, pre-examination processes, quality management systems, and a set of veterinary medicine guidance. Next slides. We also have complementary educational products that help with the implementation of these guidelines and documents in the laboratory through webinars, both live and on demand, guidance documents to help with the implementation, online courses, a certificate program in laboratory quality management systems, tools for understanding laboratory risk management, implementation of cost of quality in the laboratory, a certificate program for that, as well as EP23, online workshop for risk-based tools to meet your individualized quality control program requirements, or IQCP requirements. Next slide. CLSI crosswalks have taken the guesswork out of understanding accreditation requirements and tell you which CLSI standards will give you the step-by-step guidance you need to fulfill requirements set forth by the Food and Drug Administration, the College of American Pathologists, ISO, and you can access all of these CLSI standards and crosswalks at clsi.org/accreditation. Next slide. Eclipse is our CLSI searchable content library. We have more than 12,000 active users, with top content being accessed in microbiology, evaluation protocols, general laboratory practices, and quality management systems. Next slide. CLSI has, at the start of the pandemic, gathered a number of their documents that were relevant to the laboratory's response to COVID-19. And since March of 2020, over 4,400 copies of our free COVID documents have been downloaded. Many of the guidelines and implementation guides and the other documents in this resource are available free of charge. More than 3,300 views on our free webinars have been taken up, and this has driven a very positive impact on global pandemic testing. Next slide. CLSI is undergoing a digital transformation from written documents to access online and electronic formats. Next slide. Why should we transform? This will help with the acceleration time to market of the standards and guidelines, streamline operations in the office, will implement innovative IT or information technology platforms, help to optimize cost, and improve customer experience and engagement by increasing the speed and agility with which documents are available to the public. Next slide. So at this point, I'd like to move towards our future focus forum. Next slide. I'll remind everyone to please enter any questions that you may have in the Q&A box on this Zoom webinar platform. Next slide. The first speaker I'd like to introduce is Dr. Ren Salerno. Ren is the Director, Division of Laboratory Systems at the Center for Surveillance, Epidemiology, and Laboratory Services for the Centers for Disease Control and Prevention. Dr. Salerno, who became Director of the Division of Laboratory Systems in January 2016, has an international reputation for excellence in promoting the safe, secure, and responsible handling of dangerous biological agents in the laboratory setting. His previous position as Senior Manager for Biological Sciences and Technologies at Sandia National Laboratories included work with laboratory quality systems, biosafety, biosecurity, biocontainment, and infectious disease diagnostics. Dr. Salerno is co-author of Laboratory Biosecurity Handbook and Laboratory Biorisk Management, Biosafety and Biosecurity. Dr. Salerno is a member of the American Society of Microbiology, the American Biological Safety Association, and the Association for Public Health Laboratories. He currently serves on APHL's Biosafety and Biosecurity Committee. Dr. Salerno?
Thank you so much, Jim. And thank you to CLSI for inviting me to this Futures Forum. It's a pleasure to be here. CLSI is an incredibly important partner to CDC, and we're grateful for all the work of CLSI and all of its partners. Today, I'd like to tell you a little bit about the work that we're doing at CDC on behalf of the broad US clinical laboratory community, as well as give you some updates on what's going on with CDC and the pandemic. Next slide, please. So my division at CDC has the vision of exemplary laboratory science and practice that advances clinical care, public health, and health equity. And our mission is to improve public health, patient outcomes, and health equity through the advancement of clinical and public health laboratory quality and safety, data and biorepository science, and workforce competency. And really, the responsibility of our division is almost entirely externally facing from our agency. And we are obligated to do whatever we can in our division to support the almost 300,000 laboratories and testing sites across the country that are certified under CLIA. And so we try to do whatever we can, given our resource limitations, to support that clinical laboratory community and clinical laboratory testing in the United States. Next slide, please. Laboratory quality and safety is our mantra, is our mission, and is our focus. I'll tell you a little bit more about our work with CLIA and CLIAC later on. I'll tell you a little bit more about some of our activities around COVID-19 a little later on. I'll tell you a little bit more about our work in bioinformatics and next generation sequencing in this presentation as well. But in addition, we do a tremendous amount of work in the laboratory safety and biosafety field. In fact, if you're not already aware, I invite you to visit our lab training website, which is hopefully easy for you to remember if you're interested, cdc.gov/lab training. All of our training is free. Most of our training comes with certificates for professional certificates. And I want to point out we have new courses available for packing and shipping, fundamentals of personal protective equipment, fundamentals for working in biosafety cabinets, donning and doffing videos for PPE in relative to SARS-CoV-2. We now have 2 new training courses that are done entirely in virtual reality. And if you have the capacity and the resources to use those courses, I'm sure you will enjoy them. They focus on the setup and use of biosafety cabinets. They're available in, all of our courses are available in CDC Train, but the VR courses are also available on the international gaming platform called Steam. Over 50,000 people have downloaded the course through the Steam platform. The other thing to quickly mention about all of our courses in these broad categories are, and all of those courses specific to COVID-19 and SARS-CoV-2 are now available for syndication, which means if you'd like to be able to download those courses and into your own learning management system, that option is now available. That's a brand new capability for us at CDC. We're really excited about it. And we have many partners who've already taken us up on that. And if you're interested, please just go to our website, cdc.gov/lab training. Okay, next slide, please. The laboratory response to the COVID-19 pandemic has been challenging, to say the least. And I know it's been an incredible year for all of us in many different ways. But the nature of laboratory testing, as I'm sure all of you can appreciate, has evolved in amazing ways over the last year. You know, from a public health perspective, a year ago, say a year and a half ago, we anticipated almost all, at least most of any laboratory testing that would be related to a public health issue to come from primarily our public health laboratories and secondarily some larger clinical laboratories and perhaps some commercial laboratories when necessary. But in general, in previous public health responses, most of the laboratory testing was focused in our state and local public health laboratories. COVID has been a whole nother challenge for us, and it immediately required us to surge laboratory testing capacity into the broad clinical laboratory community. And we, as quickly as possible, engaged our commercial laboratory partners. We encouraged as many of the especially large academic medical centers with sophisticated clinical laboratories to begin testing for COVID-19 as soon as they had access to an emergency use authorized test. And then, as all of you know, we are now doing testing in very non-traditional testing locations, whether they're schools or nursing homes or correctional facilities. We're doing testing, as you know, in drive-through settings, and now we're embarking on at-home collection, which we've been doing for a couple of months, but now at-home testing is really beginning to come online. And so the testing environment has completely changed in ways that we didn't fully anticipate, honestly, and probably didn't anticipate well enough. But we've had to completely adjust how we think about laboratory testing in a public health response. And most important from my perspective is that the traditional silos between healthcare, testing for patient care only, and public health testing for public health surveillance only, have completely merged. And the traditional silos between healthcare and public health have been and now are clearly no longer sustainable. And one of our really new objectives and priorities at CDC is to really help build stronger connections across that healthcare public health traditional divide and really break down those barriers of collaboration and encourage much more work together from in the public-private space. Next slide, please. So first, real quickly, in case you're not familiar, we've worked hard at CDC to provide as much guidance to the laboratory and testing community as we can, given all the challenges of this pandemic response. These are some of the examples of the guidance documents that are now available on the CDC COVID laboratory website. Guidance specific to antigen tests, antibody tests, specific to collecting specimens, to pooling tests, to at-home testing and at-home collection, to workplace safety, to laboratory biosafety, point-of-care testing, laboratory reporting. We've really tried to cover all the challenges that we've heard all of our partners are experiencing during this pandemic, and we continue to try to be as responsive as we can, and we continue to try to invite comments and feedback from the community so that we can continually improve our own performance, just as we all take continual improvement seriously in our own laboratories. We can say that these pages alone have received tremendous amount of page views and visits. The laboratory page for CDC's COVID response is, I think, over 10 million page views. And in particular, the rapid antigen test guidance page has received almost 2 million page views since it was initially posted in the summertime. Many of these other pages also are have received over 1 million page views in this relatively short period of time that they've been up. So we know that this website and all these pages are getting a lot of attention. We get a lot of comments and questions about them, and forcing us to revise them as often as we can. But it's a real demonstration to us that the laboratory community is anxious for CDC's help. And we want to thank those of you who've helped us develop this guidance along the way. Next slide, please. The other thing that we've worked really hard at CDC to do during this response is to leverage and rely heavily on our partnerships across the government agencies, as well as across our public health partners, APHL, CSTE, ACLA, CLSI, all these professional organizations. We try to meet with them frequently as we can to get their feedback. In some cases, we will even share some of our draft guidance with these organizations for some feedback before those are posted. I hope most of you are aware that every other week we have a national clinical laboratory COVID-19 response call where we try to provide the latest information available across the government. We invite speakers from both inside and outside the government to provide sort of their latest data and information about their roles in the response. And we've also really leveraged and expanded the use of what we call LOCKS, our Laboratory Outreach Communication System. If you haven't signed up for it or opt in for those emails, we would invite you to do that. We've sent, I believe, close to 150 LOCKS messages out to those people who have opted in to those emails, and those emails provide very specific response-related, COVID-related information to all of our partners and clinical laboratories. Next slide, please. I want to just, because I know it's of interest, it's the hot topic of the day, as it should be, give you a real quick update on what we're doing in terms of variant surveillance. We are, among other things, and this is specific to the clinical laboratory community, we have engaged a number of large commercial laboratories to specifically do diagnostic testing and sequencing on behalf of the government. And so Illumina, Helix, LabCorp, and Quest are all now contracted to do, as you can see there, a large number of sequences per month from across the United States to try to get a broad understanding of the prevalence of variants of concern and variants of interest. This is just the work that is currently contracted. You can see there at the end, we are layering on additional sequencing from up to 10 additional providers in the near future. And on top of this, there's the public health laboratory system is doing a tremendous amount of sequencing as well. All that information is being deposited into GISAID to make the sequence information publicly available. And I think as of now, we've done we have collected over 30,000 sequences into our database. Next slide, please. This is the most current data on variant cases in the United States as of Sunday night, March 7th. Yeah, actually that would, yeah, Sunday night. And so we have, as of Sunday, 3,100 total US cases. Now, these cases are determined based on the guidance and perspective and decisions of the various states and how they classify cases. And these are cases of for variants of concern. You can see the 3 that are listed here. So there are obviously many other variants that are not among these 3 that are not counted in this particular total US case number. So you can, the B117 variant is the one that is sort of known as the UK variant. That's the majority of those 3,100 cases are B117 cases. B1351 variant is the variant associated with originating in South Africa. There are 81 of those, and the P1 variant is the variant associated with cases, with a variant that was first identified in Brazil, and that has only 15 cases in 9 jurisdictions. Next slide, please. The other thing I wanted to really quickly touch on, because I think it's fascinating and frustrating and difficult and challenging, and will focus a lot of our attention going forward, is the challenge that we all have had around test result reporting during the pandemic. And again, as I mentioned earlier, the challenge here is that honestly, we just didn't anticipate the need for such wide-scale testing by so many different types of providers. As I said, if only the public health labs need to report diagnostic results of a agent of public health concern, the reporting is fairly straightforward. But as soon as we had to surge to not only traditional moderate and high complexity CLIA laboratories, but drive-through collection, point of care testing, non-traditional testing sites, now at-home testing, the reporting became really, really complicated. And you can see this is a very simplistic graphic of all the different organizations and all the different ways that they're reporting their test results. And we have heard and appreciate that this reporting mechanism has been extremely challenging, frustrating, and difficult for many organizations, and is really made, frankly, surveillance for SARS-CoV-2 challenging as well. Next slide, please. This graphic is where I'm hoping that we can go in the future. And this is something that we had done a little bit of work on pre-pandemic, and we now realize we need to do a lot more work to advance electronic test ordering and result reporting. And we call that eTour. And what it is the creation of two-way systems between clinical laboratories and public health laboratories, and that those systems enable seamless communication and reporting between the commercial clinical labs and the public health lab in their jurisdiction. And so that there's no need to send emails or faxes or even to ask many questions about what needs to be reported to whom, that all this data could just seamlessly stream from the clinical laboratory community to the public health laboratory community through existing cloud-based IT systems. So we really hope that we'll be able to build this two-lane highway. The idea here is that in addition, it's not just one-way communication of lab results going from a clinical lab into public health and then the clinical laboratory community really getting no feedback or information. But we want a two-way highway that enables the public health labs to provide information of relevance back to the clinical laboratories. The best example is newborn screening. And in some cases, these eTour systems for newborn screening already exist. But unfortunately, in many cases, the clinical labs, hospital labs are still ordering newborn screening tests by email and getting email results from public health laboratories, which we've got to change and improve. And so this is what we're going to aim for going forward from a reporting point of view. Next slide, please. All right, now I'm going to move fairly quickly. I'm hoping that most of you are familiar with the Clinical Laboratory Improvement Amendments of 1988, or CLIA. It's a tri-agency program in the government, and CMS regulates the laboratories, FDA regulates the devices that test the assays, and CDC is the medical and scientific advisors to the CLIA program. Next slide, please. CDC's role is, as I said, as sort of the scientific and medical advisor. We identify technical and scientific issues in clinical laboratory medicine that need to be addressed. We develop and distribute that information and educational materials to the community. We manage CLIAC, but we really serve as essentially, we sit between, if you will, the regulated labs on one side and the regulators on the other side. And we try to manage that relationship on behalf of the entire laboratory community. Next slide, please. CLIAC is a advisory committee, a federal advisory committee that meets twice a year that helps the government, meaning CDC, FDA, and CMS, understand what changes need to be made in the rules and regulations, and helps communicate what challenges are happening or technical advances are happening in the laboratory space that the rules and regulations don't adequately appreciate. Next slide, please. This is the way. So CLIAC advises on CLIA. That advice goes to the three agencies. CDC manages the committee, and then CLIAC provides and can also provide a recommendation to the HHS secretary if it wishes. All those members are appointed directly by the HHS secretary for terms of 2 years. Next slide, please. This is our most recent CLIAC meeting. It was the first time that we had an entirely virtual CLIAC meeting over Zoom. So we also were learning how to Zoom a federal advisory committee, which was a first for us. We will do that again at our next meeting in April. Next slide, please. And then I put this slide in here too, because this is something where, again, we need the laboratory community to help us. But one of the things that I want CLIAC to be really thinking more and more about, and COVID has brought this to the fore as well, is what new technologies are increasingly becoming part of the clinical laboratory community that really make sort of traditional approaches to CLIA much more challenging. So the best example from my point of view is the, the traditional definition of a laboratory as 4 walls and a floor and doors, and you enter the lab and then you exit the lab. And everything inside the lab is CLIA certified and everything outside the lab isn't. That kind of definition has really gone away. The fact that we're doing so much remote work that includes laboratory work, laboratory analysis, is really important for CLIAC to understand and for the government to understand, to be able to adapt our rules and regulations to accommodate all these changes in technology. Next slide, please. Our next CLIAC meeting is in next month. It will be virtual. Please visit the CLIAC website for more information about this meeting. I think it's going to be great. The theme will be clinical laboratory medicine in the age of COVID-19. We're going to be talking about clinical laboratory perspectives on laboratory developed tests, application of CLIA regulations during the COVID-19 pandemic, and how those have changed, and how they, whether or not some of those changes should be permanent, or whether we need to return to some degree of CLIA normalcy. As well, we're going to focus on the expansion of point of care and at-home collection and testing, and what that does to the way we think about CLIA and quality and safety in the clinical laboratory environment. We think it's going to be a great meeting. We hope that you'll join us virtually. And I believe that is it for me. Thank you very much.
Thank you, Dr. Salerno. I would remind our audience, considering Dr. Salerno's presentation, how can CLSI help laboratories move forward productively into the future? So how can the government, what do the laboratories need from the government perspective, and the things that were brought out in Dr. Salerno's presentation? If you can respond to that in the chat box, that would be appreciated. Next slide. And please, reminder to enter any questions into the question and answer box. Next slide. At this point, I'd like to turn to the healthcare professions perspective and Dr. Gary Prokop. He is the Director of Molecular Microbiology, Virology, Mycology, and Parasitology at the Cleveland Clinic. He is past chair of the Departments of Clinical Pathology and Molecular Pathology, past section head for clinical and molecular microbiology. He's a diplomat of the American Board of Pathology and Anatomic and Clinical Pathology and Medical Microbiology. He's a fellow of the American Academy of Microbiology, the College of American Pathologists, the American Society for Clinical Pathology, the Infectious Disease Societies of America, and the Royal Society of Tropical Medicine and Hygiene. He's a trustee and past president of the American Board of Pathology. He's given more than 615 scientific presentations, has 204 published manuscripts, 49 chapters, and 3 books to his credit. His primary interests are developing and promoting best practices in laboratory testing, the practical applications of molecular diagnostic methods for the diagnosis and treatment of infections, infectious disease pathology, mycology, and parasitology. It's my pleasure to welcome Dr. Prokop.
Thanks so much, Dr. Nichols. Can you hear me fine?
Yes, we can.
Okay, great. Thank you. Thank you for that wonderful introduction. And I was asked to look into the future and see what will be important for the professions. And I'm going to start with a disclaimer from Yogi Berra. It is tough to make predictions, especially about the future. So I've done my best. If we're way off, you'll know why. But I think we'll be pretty close. And I was pleased to see that many of the topics from Dr. Salerno and I were similar. So one of the first topics I'd like to talk about that I think is going to be, it's big now, it's going to be much bigger in the future, and that's the idea of improvements in coordinated care. This idea, this old idea that you get sick, you have acute disease, you go to the hospital, you go home, you go to a nursing unit or a nursing care facility, and you're kind of a hot potato. That patient is. They're the responsibility of where that patient lands. That idea is changing in healthcare across the nation, and you'll see a number of the drivers in that. But this is really an idea of team-based care delivery. You can see one of our integrated care model images on the right. Everybody is responsible for that patient in this system, regardless of whether they happen to be at home, in a hospital, in a physician's office, in the emergency room, etc. We're responsible no matter where that patient is. I'm going to give a pull-out example from this in just a minute on our rapid responsibility. We've seen the importance of rapid response in COVID-19, but it really goes beyond that. And then really optimizing our inpatient care services from this idea of centralized monitoring, where multiple ICUs can be monitored in one location, to some of the blocking and tackling that we know is needed to improve care delivery, like continue efforts to decrease hospital-acquired infections, continued efforts to decrease readmissions. And again, much of those efforts will go back to this entire integrated care model. We'll get better if we look at the patient's issues as our issues, no matter where they land in the system. And so this has much to do right now, and again, we'll have more to do in the future with this coordinated placement, whether the patient goes home to a long-term acute care facility, to a skilled nursing facility, and we really need to have appropriate follow-up to help decrease readmissions. A quick example, one of the biggest causes for readmissions is congestive heart failure. There have been hospital systems that have built scales. When people start taking on water, that's when their congestive heart failure will decompensate. The scale will send a message to the healthcare providers, and they will intervene so that patient can be treated appropriately in the home or where they are without having to be readmitted. Just a good example of this. Next slide, please. This is an example of our mobile stroke unit, which has won awards, and we're really proud. I'm not going to go over, you'll have access to these slides. I'm not going to go over, but this is an idea of taking it to the streets, right? The idea of the physician with the little black bag is coming back. The little black bag has gotten much cooler, as you can see in these images, with taking everything you would need to respond to a patient with stroke. As you know, stroke is a time is of the essence disease. And you can look and see the statistical differences on time to appropriate therapy, etc. And this is really translated into better outcomes for patients. So in some situations, it's better for the patient to come to us. In other situations, it's better for us to go to the patient. And that may be something like a stroke, or it may be just for assuring, chronic care management is being done like it should be done. Next slide, please. Looking into the future, there's got to be changes in healthcare economics, right? There needs to be better alignment of the cost of care, and we're seeing that more. We'll see this in the next slide also. Better alignment between payers and providers, getting on the same page. There's a lot of money lost in supply chain. If you're in a hospital system and you don't have a centralized supply chain, you are losing money in health economics because vendors are taking advantage of that. There's opportunities to reduce waste. We've actually seen this for years, but there's more opportunities in the future to introduce, where appropriate, and I'm going to underscore where appropriate, industrial standards into laboratory medicine and other areas in healthcare. Pre-authorization, and again, this is going back to that alignment of cost of care, is unbelievably frustrating to I think everybody involved, to physicians trying to give care, to patients trying to be cared, to payers who are afraid that they are being misused and abused by folks who are inappropriately using the system. What we have now is we have a system of denials and adjudication of denials that go back and forth, huge amounts of waste. When we can get on the same page with regard to optimal utilization of resources, and again, that's alignment of the cost of care, much of this waste and cost can go away. An example would be the empowerment of PharmDs and the prescription drug costs. These are folks that we have really, and you'll hear this theme of empowering people to function at the top of their license. And PharmDs are now doing things on the floors that would never have been thought of 10 years ago. And guess what? Our healthcare delivery is much better for it. So there are many opportunities that we haven't explored yet. Look at the graph to the right just for a moment, and look at the United States. We're number one, right? Yeah, we're number one in healthcare expenses as percentage of GDP. It's not something we should be proud of. We're going to show you another slide on the next slide. Go to the next slide, please. And this is this migration, and these are in sync, hand in hand, from a volume-based care delivery system to a value-based care delivery system. Not only are we spending an unbelievable amount, health spending per capita on the x-axis, but look at our life expectancy compared to other countries. Many other countries are spending a lot less and have better patient outcomes, better health than us. And I know we're a prideful nation. This is a time to put that away and be introspective. There are ways to actually spend less and have better outcomes. And I think that's something we're going to have to look at hard in the future. Some of this is going to deal with population health management, honestly addressing issues of health disparities. There's huge opportunities, and we learned this through COVID-19 and all of our Zooms, right? You can do distance-based health to individuals who can't come to a provider. And again, this idea of having individuals function at the top of their license. Really embracing, and maybe in some cases almost mandating, preventive care, higher vaccination rates, or promoting early screening, early diagnostics, excellent, outstanding chronic disease management. Again, other ways to get out to patients who are maybe not managing their diabetes appropriately, et cetera. And then truly addressing medication costs. You cannot undertake initiatives to better manage chronic disease if patients are splitting their insulin in half because they can't afford it. So, but these need to be addressed hand in hand with this idea of decreasing waste while increasing efficiency and preserving or enhancing value. And we've demonstrated that can be done over the years in our laboratory stewardship test utilization service. Next slide, please. Looking into the future, what else are we going to see a lot more of? You already heard about next generation sequencing for microbes. We'll get there in a minute. That is also in next generation sequencing of humans, right? So genomic medicine. And so following Moore's law, which is really written concerning the doubling of computing power every 2 years, this is flipping that coin, and this is decreasing the cost of sequencing by half every 2 years. And you can see that in the green dots. So it is becoming more cost effective to just sequence the whole exome or somebody's genome than going after individual genetic targets. Maybe not there yet, probably will get there in the future, where you're going to have an entire genome to examine along with the history and physical when somebody comes into the hospital. Now that's a big deal for a variety of reasons. What if somebody orders a whole exome sequencing because they're interested in hearing loss, but in that data set, there are also genetic predictors of neurodegenerative disease? How do you handle that? Needs to be dealt with. In our genetic assessment, we will have variants of undetermined significance. What happens when we determine the significance of those variants 5 years from now? Well, we have to have a way to go back and contact those patients, because 5 years ago they were told, you know, we don't know what this variant means, probably nothing. Most of them are probably nothing. Now we know it's something. You get the idea. This is going to be a substantial investment in informatics, and there are risk management, risk benefit management aspects that need to be considered. So something in our future for sure. Next slide. So there's a bit of a debate on how many cells in your body belong to you and how many belong to microbes. And I won't weigh into that, but you got a lot of microbes in your body, either as many cells as belong to you or more. And it's really not surprising. A great majority of these belong in live in your food tube, right? In your intestinal tract. And what does your intestinal tract do? It absorbs things. So as those microbes are metabolizing, it really is not a far stretch to think that you are absorbing some of those metabolites. And those metabolites have associations with health and have associations with disease. And this is a burgeoning area. Much written already, but much more will be written in the future. And there are already papers and discussions on how to modify your microbiome. And I'm not talking about taking antibiotics. I'm talking about understanding the metabolic pathways of the organisms that are present in your body, so that you can do things that would be beneficial to health and would decrease disease. So pretty interesting stuff. Kind of gross and unbelievable how successful fecal transplants have been in folks with recurrent C. diff disease, just as an example of modification of the microbiome. Next slide, please. So infectious diseases, and we've seen this picture way too much. That's absolutely sure. There's more to it than an emerging pathogen response, my second bullet point, and you've already heard a lot about that. And I think we have learned a lot from both things we've done well and things that we could have done better. And so, you know, the teams you've heard from already and others, I'm sure will take this to heart, and we'll have a better emerging pathogen response in the future, improved. We also have to think about things like emerging antimicrobial resistance, always there, and needing enhanced antimicrobial stewardship. You know, great incentives for us to do this as we whittle away at our effective agents. So we need to preserve what we have, and also get more of agents that can be used to address infectious diseases. As we get better and better at keeping people alive and making them healthier, transplantation, the use of biologic treatments for autoimmune diseases and the like, we have increased rates of other infections, right? Transplant-associated viral and fungal infections, increased incidence of tuberculosis, histoplasmosis in patients who take biologics, and you know the opportunistic infections in patients that are neutropenic because of chemotherapy or C. diff disease, chemotherapy and antibiotics. So again, all issues we are addressing now, but will continue to be issues as we look into the future. Next slide, please. So basic informatics, and so and I was pleased to see these were also on the previous speaker's slides. We are using these clinical decision support tools to optimize care right now. They're pretty unidirectional, just telling or stopping a provider. We probably need to make these less interruptive and more personalized. But they do have great possibilities. We can stop life-threatening events. If somebody was going to give someone with a penicillin allergy a penicillin, we could stop that order in its tracks. The bullet point above that, with regards to individualized, we could look at individual patients' renal function, and there could be recommendations or adjustments to medication levels based on their renal function. In the future, I think, and I hope, we're going to see more algorithmic testing. We have known for many, many years, I talk to, when I give some of these talks, we've known what tests that need to be ordered for hypothyroidism for many, many years. Why are we still ordering those things a la carte? We can build diagnostic algorithms that can address most commonly occurring diseases, and that should be done, and it would decrease a lot of the waste in the system. Next slide, please. Okay, artificial intelligence and machine learning. I like and I don't like this picture. I like this picture because a lot of people are threatened by artificial intelligence and machine learning. This looks like a battle, arm wrestling between a man and a robot here. I would love to see a picture where they're shaking hands, because I think that's where our future is going to go, where we're going to utilize artificial intelligence and machine learning, not be threatened by it, to become better healthcare providers. Some of the questions we're going to have to wrestle with are, one is going to be validation. How do you validate an iterative learning process? This thing is going to get better every time. So it's not like you're validating something that's said and done. It hopefully will never be said and done. Got to answer that question. Is this a laboratory developed test, or is this a product for FDA approval? And remember, if it's FDA approval, it's iterative. How is this incorporated into care delivery, and who's responsible for errors? Who is giving the care? So these are points we're going to have to wrestle with. All questions here, no answers from me, but I look forward to hearing from people smarter than I am. Next slide, please. Okay, big data. Along with that, this idea that we have reams and reams of data that are not being accessed. And I'm going to point to the folks at the Santa Fe Project and Lab 2.0 and how they're using so much of this data comes through the laboratory. I think this is a huge opportunity for pathology and laboratory medicine to get into more of the healthcare delivery business, and to use laboratory data to drive population health management. And look up this group if you don't know them already. They've done some wonderful things. The next is using this data to predict things, pulling data from multiple sources and finding patterns that may not be obvious. There are groups that are thinking about and working on this already. I've had one contact me wanting to do research, and I won't use any names, but they said, have you ever been on an airplane where it was forced to land and nothing was wrong? And that was because sensors started going off that saying things in the engine were about to fail. And they're looking into the healthcare space. Can they pull all this data to predict what might happen or what's going to happen before it happens? I heard a wonderful talk from the chair of one of the Harvard hospitals, whose folks published some papers looking at how they could predict who would have anemia based on parameters from normal CBCs. So pretty interesting. Next slide, please. And I think we're rounding it out here. Medical education really needs to change from this traditional time-based approach to a competency-based approach, wherein we know that a doctor or other provider can do X because they've demonstrated competency, not just because they've answered some multiple choice questions and been through a training program of X period in length. So much to do there. Our Canadian colleagues are way ahead of us in competency-based care, but we will catch up. So I believe that's my next slide, and I will turn it back to the moderator.
Thank you, Gary. I'd remind everyone considering Dr. Prokop's presentation, how can CLSI help laboratories move forward productively into the future? Please respond in the chat box. Next slide. And please enter any of your questions that you may have in the question and answer box. At this point, I'd like to introduce Dr. Darlene Solomon. She is Senior Vice President, Chief Technology Officer for Agilent Technologies. Her responsibilities include Agilent Research Laboratories, which focuses on high-impact longer range research in support of Agilent's sustained business growth and Agilent's programs in university relations, external research, and venture investment. In her leadership role, she works closely with Agilent's businesses to define the company's technology strategy and R&D priorities. Solomon serves on the board of directors at Materion Corporation. She also participates in multiple academic and government advisory and review boards, including current and recent appointments to the National Academy's Board on Chemical Sciences and Technology, Visiting Committee on Advanced Technology of the National Institutes of Standards and Technology, Stanford Interdisciplinary Life Sciences Council, UC Berkeley's College of Engineering Advisory Board, and the Singapore National Research Foundation's Scientific Advisory Board. If you can welcome me now, Dr. Darlene Solomon, on the industry perspective of the future of the laboratory.
Thanks so much, Jim, for that introduction. Let me know if the audio is not coming through. And I really want to thank CLSI for the incredibly important work that all of you do throughout the year. And I am delighted to have the invitation to join you today and contribute to the look to the future with some industry perspective. So moving to the next slide, I thought I would highlight, I started with a brief introduction to Agilent, but then highlight a couple of the areas that I think are really transitioning in their clinical utility, as well as highlight a few areas that I believe will be beneficial to be on the radar screen for CLSI. So moving to Agilent, and the next slide. So our products and solutions help improve quality of life, serving analytical and clinical laboratories throughout the world, 275,000 of them. And that focus is one that is across 6 different markets, what we call the applied chemical markets, areas like food safety, environmental, air and water quality, chemical energy. Our largest market and customer focus is biopharma, and our increasingly important focus over these last several years has been really taking our capabilities more and more to advance clinical research and clinical diagnostics. And of course, academic and government research, which underlies all of these areas. And so, in the analytical lab, it's very, very focused on chromatography, mass spectrometry, spectroscopy solutions, full workflows, including sample prep and informatics, both incredibly important. And then reagents and solutions for life science research and clinical laboratory, very much our focus with today's event. And in particular for Agilent in cell analysis, pathology, and genomics. Turning to the next slide. I very much believe that these are what we will look back on 30 or 40 years from now as the early days of the century of biology. And this century will take decades, but very clear that the first wave is precision medicine, which is our ability to understand and treat disease at a molecular level. The range of technologies, the range of source materials, and different types of analyses that are all part of this molecular understanding of disease are really kind of illustrated in this complex diagram, and it is complex. That's why it's going to take a century. But many believe it will be possible to really be able to do this, to not only, I'm going to say, go beyond the powers today, but to ultimately understand the pathways and engineer and predict biology the way we do the physical sciences. Technologies and disease understanding are very much moving through this continuum from research to translation into clinical research and clinical diagnostic utility. But clearly, standardization is critical to obtain the trust and reproducible results required for this outcome. So the first example on the next slide moves us to clinical mass spec. And this is an area which includes many different levels of molecular understanding. Mass spec helps us do the conformational identification of molecules to a very large extent, not 100%, but very, very strong. I won't go into that further, but lipids, carbohydrates, oligonucleotides, things that are not easy to characterize and very complex are measured by clinical mass spec. And I think that it's a very powerful contributor to advancing clinical research that is poised for greater contribution to clinical diagnostics through regulatory approval and adoption. As shown on the next slide, there's a number of areas that are already being addressed by mass spectrometry, driving clinical results and improving patient outcomes. Liquid chromatography and gas chromatography combined with mass spec are already gold standard in a number of areas like clinical toxicology, therapeutic drug monitoring, vitamin D and metabolite analysis, inborn errors of metabolism, and microbial identification. But looking forward, some of the emerging applications include vitamin panels and correlation to disease, endocrinology, multivariate analysis, often omics panels that bring together genomics with metabolomics or metabolomics with proteomics in order to drive diagnostic and prognostic results. And we'll come back to that a little bit later. Spatial imaging, especially for tissue and sampling off of surfaces, is an emerging important area for biological and clinical understanding, perhaps diagnostics at some point. And we're already seeing emerging biomarker opportunities in COVID-19 and infectious disease, predicting the severity of human response to better inform clinical care. To the next slide, there are challenges, however, in moving mass spec to the clinical lab. Technology robustness compared to traditional clinical analyzers is a challenge for mass spec. The variability, especially introduced by sample prep for complex matrices. The mass spec may be very reproducible, but the entire workflow needs to have clinical outcomes, and sample prep is a key area. Ease of use in the clinical lab, harmonization of results across labs, between operators, and obviously moving forward to meet regulatory requirements in the US and globally. Advances in the industry are ongoing. That is required. It's absolutely important to Agilent. You see some of our platforms here, but you see it throughout the industry, seeing the opportunity of what mass spec can uniquely bring forward. So platforms are becoming more robust, more reliable, more, I'm going to say smaller footprint. And especially important is automated sample prep instrumentation with standardized intuitive user interfaces. We can't have the skill level being above what is acceptable in these types of more broad, comprehensive environments. And the sample prep, because sample prep isn't just about when it's automated, it's not just about high throughput, but it's really about reproducibility and being able to remove a lot of the manual variation that is critical in these assays. Software development and informatics, as we've discussed, and then the development of internal label standards to drive calibration and quantitation, and the need, again, for workflows, non-expert users, more and more validated, decentralized kits that can lead to standard results outside of the main laboratory, and of course SOPs and work instructions. The next slide just gives one example of many where mass spec can provide unique clinical value, and this is in some of our collaborative research with Baylor College of Medicine and Drs. O'Malley and Srikumar at Baylor. And this study is aimed at non-invasive early cancer detection, prostate cancer, and prognosis in African American men. And there's a disparity in prostate cancer, which is found to be 60% more common in African American men than in European American men. And generally, these individuals have higher tumor volume, PSA levels, and levels of recurrence. And so with these changes, the questions are whether there are metabolic alterations that can be measured and provide further insight into patient care. And so this clinical research aims to develop high throughput LC-MS methods based on metabolomic signatures in plasma from blood for early detection and prognosis of prostate cancer. And the research has identified initially 26 biomarkers identified through clinical biopsy cohorts of these African American and European American individuals. And these studies often start with like 100 individuals and then move to several hundred and so forth as they go forward. But when you're looking at something like 26 biomarkers that are covering, multiple biological pathways, it's something that becomes more difficult to do. And it really plays to what mass spec can do well compared to something like an ELISA, which is great for a handful of analytes, but not when you get to 20 or 30. So in taking something like this forward, of course, there's many considerations to move from research to routine. Obviously, identifying and validating the right biomarkers to answer clinical questions and provide clinical actionable utility, getting the right patient population cohorts for validation, the automation for workflow reproducibility, and the deep understanding of regulatory guidelines to help translate these biomarkers into clinical use. Moving to the next topic, the second area I wanted to highlight is immunohistochemistry in companion diagnostics, where there's also a need for increased standardization and harmonization. Companion diagnostics are used to select patients who are most likely to benefit from a particular therapeutic. And as such, they're developed together with the therapeutic in terms of early phase clinical trials, and then submitted together for regulatory approval. Next slide, please. So why standardization of companion diagnostic assays is important is shown with some immunohistochemistry examples here. So if we look at, on the left, a typical class 1 IHC diagnostic, it's really about confirming a diagnosis. It's a qualitative test that measures the presence or absence of a biomarker. And minor sensitivity variations between lots of the antibodies is more acceptable because the overall test outcome will not change. So as illustrated here, whether there's 2, 3, or 4-fold presence of that marker, it's a carcinoma or it's not present. It's not a carcinoma. Perhaps a different tumor type. In contrast, as we look to the right-hand side and a class 3 companion diagnostic, it's still qualitative, positive, negative, but its action revolves around a given cutoff. So we're the goal is to select patients that will most likely benefit from a specific drug before it's actually provided as a drug. And the efficacy is established based on a threshold, a cutoff point, so that only patients who express a certain amount of the biomarker are eligible for treatment. And so assay lots and interpretation must demonstrate the same sensitivity at all times across sites. And so you can see here where the top panel is plus 400 positive, it's above the cutoff, clearly eligible for drug therapeutic treatment. At 300, it's borderline and should be retested. And this is for subtest, by the way, as the example. At 200, it's negative, it's below the cutoff. And of course, at 0, it's negative and below the cutoff. In both cases, not eligible for drug treatment. On the next slide, as we think about quality control, quality assurance for design control and implementation, the idea is to perhaps think about where recommendations can go to the next level for quality assurance in these IHC companion diagnostics in light of their inherent higher complexity and higher risk to patient management. So CLSI, of course, already has very strong IHC recommendations in place for quality assurance of various aspects of the assay for ILA 28-82, and those are listed here. But looking forward, there are additional trends and opportunities worthy of some consideration going forward. And that includes the continuing increase in biomarker-driven development, which is very, very prevalent in clinical research today. It's really about getting it to the next level of being able to have full clinical approval and use. still a barrier, but moving forward in higher throughput studies. The other thing is reduced sample size. They're getting smaller and smaller, especially with biopsy samples. And there's more and more molecular information that's being desired, certainly on the genomic side, but I think that's going to happen also in terms of other areas like metabolomics and proteomics as well, especially depending on the disease area. Standardized assay protocols and controls for consistent and effective clinical use. Once again, big role and big opportunity for digital pathology, which is moving forward, enabling increased quantification and assay complexity because of its capability to see beyond the human eye. And of course, we all know how digital pathology has accelerated this past year with COVID. There are multiple tumor indications with different interpretation in terms of being able to look at a broader set of biomarkers to understand immune cell components, to understand areas like tumor microenvironment. I'll come back to that in one of our end examples. And multiple assays are available associated with different therapies for some biomarkers that are based on different antibody clones. And a great example here is PD-L1, and we'll take a closer look on the next slide. Using PD-L1 assays helps to illustrate some of the challenges here. So pictured are a few examples of PD-L1 assays approved in Europe and US. There's also many examples of assays and standalone antibodies that are gaining approval worldwide, including China and Japan. And these various approved tests have different antibody clones across different tumor indications and different scoring interpretation. And all of this is tied to clinical outcome data, and we want to ensure the best assay results for correct scoring and diagnostic cutoff. Looking here at these examples, there's been multiple studies published that show there's interchangeability between these first three assays. So the PD-L1 with IHC 28.8 and 22C3 and SP263 out of Agilent Dako and Fontana Roche, they've been evaluated and find that there's quite good interchangeability, but that the SP142 has been shown on several occasions to be a consistent outlier. Ultimately, the clinical utility of these PD-L1 assays has to be evaluated for its predictive value, and additional studies could be designed to better understand this variation, stain PD-L1 tumor samples of patients treated with immunotherapy, for all four of these assays, for example, and additional ones, and comparing their predictive value. Being able to develop standardization through robust and high utilization of proficiency testing would be a direction to consider, as well as the use of digital pathology and AI, as you've heard earlier today, to better standardize differences in assay intensity at a higher resolution as far as interpretation. And also there's some great work that's underway showing how digital pathology can lead to better reproducibility and training across pathologists. Again, the benefit is all around ensuring the assay results are tied to the correct scoring, diagnostic cutoff, and treatment option for the patient. Okay, in the last few minutes, I'd like to just highlight a few additional areas. These are ones that are already important today, and I believe they're going to be increasingly so looking forward. So the first is highly multiplexed, as shown on the next slide, highly multiplexed spatial, cell, and tissue analysis. And to illustrate, I've borrowed a slide from Akoya Biosciences. While conventional IHC allows pathologists to see one cell at a time, the vision of spatial omics is to permit researchers to combine highly multiplexed RNA and protein quantification with morphological analysis to generate unique information about the tissue biology. And this is especially important in areas like immuno-oncology, where tissue microenvironment is appearing to play a very key role in the patient outcome. So this shows, again, just two examples. There's many other companies out here doing great work, but they have the Codex assay, which is more for the discovery side, the clinical research, and then Phenoptics, which is more for translational studies, fewer markers, but higher throughput. But in the discovery side, the ability to actually look at like 50 markers a day in a few samples. And so DNA barcoded antibodies are combined into a cocktail. They enable the full tissue to be stained in a single step, which is a pretty simple workflow, and enable pathologists to investigate large numbers of cell types that can reveal these cell-to-cell biological interactions that will relate to drug response. And of course, you need very complex software to make all of this simple. Second area is liquid biopsy. This is liquid biopsy has tremendous promise. It's getting major investment from all around research labs all around the world and many, many different companies. And the tremendous investment is based on the tremendous promise. Being able to do blood-based analysis, of course, is less invasive. You can work across tumor types. And looking forward, one of the most important opportunities is that once you remove the resect the tumor, you don't have something to measure, but you can continue to probe for therapy response monitoring and recurrence metastasis, potentially through blood. I think the holy grail is the area across the left here, which is risk prediction and screening, but much more approachable and on the horizon is the use of liquid biopsy for diagnosis and therapy selection. And as I say, for then looking at the patient trajectory going forward. Multiple modalities, techniques, and approaches towards liquid biopsy. Cell-free DNA or circulating nucleic acids is one major category. The other is circulating tumor cells, which would look at potentially DNA, RNA, protein, and so forth, and other forms of exosomes. I think that there are, though, still many, many challenges. It's early days, but this is going to become increasingly important for all of these reasons. But again, standardization, protocols and techniques, understanding thresholds, understanding what's real versus what's extraneous at very, very low levels of detection, and of course, quality control standards. The last area is cell and gene therapy programs. And cell and gene therapy are really, for me, in many ways, this next big wave of biopharma, going beyond monoclonal antibodies and oligonucleotide therapeutics to really introduce capabilities that can cure patients. In our look at where the current programs are in preclinical and clinical phases, we project that actually by around 2022, maybe just a little over a year from now, the number of programs based on cell and gene therapy is forecasted to actually surpass those of antibody biologics. And that with about, I think, around 2,700 of each at various stages of development. In terms of challenges in cell therapy, they are absolutely significant, but just to give you an idea of the complexity, the first here, functional potency, requires stringent quality control in cell therapy. A single quantitative biological assay is not likely going to be sufficient. Similarly, the challenges of predicting in vivo performance, translating how in vitro results of how that therapy will actually perform in a person based on animal models or in other patients. And, you know, cell therapies in terms of manufacturing are just a whole nother world compared to small molecule biologics or antibodies. Not batch and scale like traditional bioreactors, especially for autologous, much more individual. And, you know, the cell itself is the drug. And these are living products. They don't just kill cells, but they also proliferate. And that's an important part of the mechanism. So introducing many, many new types of consideration. I think this is an area with a very, very long runway, but a very exciting one that is really gaining traction quickly. So with that, I'd like to, I think that's my last slide. Yeah, I want to thank you for, again, the opportunity to join today, and be glad to try and answer any questions you may have. Thank you.
Thank you, Dr. Solomon. So I remind the audience, if they would like to consider Dr. Solomon's presentation, how can CLSI help laboratories move forward productively into the future? And there's been a couple of suggestions that have been put into the chat box. One of them is to think about real-time panels, just like this one, for hosting questions, sharing information on emerging topics. Another idea that was brought forward was the future state of the reporting structure being very crucial, coming out of the government perspective. The second, from a point of care aspect, to take more reasonable requirements for point of care and to help take into account new technologies. We have on the questions side, on a global basis, CLSI is viewed as too expensive. Can CLSI find other models to fund documents, work other than document sales, and provide free documents so the work is not only a country club benefit? So I will take this one on. I did, in one of my slides, talk about the global partnerships of CLSI. This is through grants where we work with local contractors. And it is not just here, we're providing you guidance documents, now go with them. It is actually directed through workshops, through pre-measurement of where the quality is, what particularly needs to be addressed in that laboratory in terms of quality management systems, and directing our documents to those aspects of the laboratory. And then as follow-up, coming back several weeks and months later to help assist with the accreditation of those laboratories. So it's providing the documents as part of a package deal. There is also available now several free documents on the web to assist labs during the pandemic in testing and improvement of quality for COVID. So there are different aspects of the documents, and they are being provided through different programs. So I hope that addresses that question. In the last minute or so, I want to open it up to each of our speakers to provide maybe their top follow-up with this. And maybe I'll ask Dr. Salerno, do you have one last follow-up statement?
Thank you. My quick follow-up is I really appreciate the comments and the questions in the chat, really hitting on a couple of the key issues that we need to continue to work on from the federal level. And, I promise to try to do the best that we can to make some substantive progress on those issues in the near term. Thank you.
Dr. Prokow?
Yeah, interestingly, one of the questions in the question and answer, someone mentioned about entrustable professional activities for education. And, I don't know that CLSI has ever looked at standards for education, but as we move to competency-based education, it may be a new space for them.
Great, thank you. And Dr. Solomon?
Thank you. I would probably just kind of reiterate that there's a real inflection point in terms of moving to a molecular level understanding of disease, and it holds great promise, but it really introduces a lot more complexity and a lot more variation that needs to be understood and controlled.
Super. If we could have the last slide. I would like to wrap up by just providing everyone with some helpful CLSI links. CLSI is available at clsi.org, and you can also reach standards, global training there, as well as the COVID resources. I want to thank you for your attention to this Future Focus Forum and hope that the information that's been provided will be useful for you in your everyday laboratory work. Thank you.